The patient is put on a machine, and or the donor, the blood is being pumped out into a cell separator. There's a blood thinning drug is being put in to make sure the blood does not clot, and then the cells you want is collected in a bag. The rest is go back to the person. The principle is amazingly simple, but we have been using this for about in the hematology field for 30, 40 years. Initially, it was used for totally different reasons, but now it's also used for collecting hematopoietic stem cells. But I want to emphasize a couple of points. One is that it's based on G-force, and then instead of a tube, you're using a plastic type of bag. And the second point I want to mention is that the stem cell is collected in this layer of the this layer of the cell column, what we call the white cell column, and therefore it's not totally purified. All right, so it's not completely purified peripheral blood stem cell we collected using the G-force. Come back to this patient. She had a autologous, means getting from her own cell, transplant in October 2002, and 8 years down the track, she has no more scleroderma and is back to full-time work. Now, as people say, it's only one case, so what? I think that's very important in clinical trials. We have found that now, up to date, majority of our patients have good outcomes, either resuming work or study, but it's not all are successful. And that is why currently there's an international randomized study looking at the benefit of or otherwise of this treatment, and hopefully we'll get an answer by the end of next year. Now, the next example is that in the totally different area of applying hematopoietic stem cell for treatment of another condition, what we call ischemic heart disease. Ischemic heart disease is because I think we enjoy ourselves too much, eating too much fatty food like McDonald's. And I'm sorry, anyone who's, I hope we're not sponsored by McDonald's. And you can see there's a narrowing of the artery here. The problem is that you're not just dying from sudden heart attacks, because you can actually get to very severe chronic heart failure. And at this stage, one of the only options available for end-stage ischemic heart disease leading to heart failure is heart transplant. So, in the last few years, a lot of animal studies been done using stem cell therapy and suggest it might be benefit. So we and the Victor Chang Cardiac Stem Research Institute in the last few years embarked on a study of using peripheral blood stem cell to treat this type of end-stage heart failure due to ischemic heart disease. How many more minutes I have, Robert? 4 minutes. Thank you. All right. Okay, so I have to rush through it a little bit. And basically is that the design of the clinical trial is in the two parts. The reason I want to mention this is that the design of the clinical trial or clinical trials are very important, because that will have a major impact on the outcome of the results or the findings. We divided this into two parts. One part is just giving G-CSF. Remember the protein that we mentioned before that can mobilize blood cells. The second part is that we give the G-CSF as well as the stem cell, collecting the stem cell, and then give the stem cells into the coronary artery directly. We either give it with purified forms. You remember I mentioned before, using a cell separator is unpurified. And now we say, okay, we want to ask the question, maybe it's the unpurified cells that are giving a benefit, not the purified stem cells. And that is why we split this into two groups, blind study, purified stem cells. And then assess the patient every single step to address the question whether this has any benefit, this one has any benefit, or this one has any benefit. Okay. A bit about technology. The simple technology of the machine is that a cell separator based on two principles. One is that is the antibody antigen affinity, and the second one is the magnetism. Right? And this is because it's a nanomedicine meeting, I thought I got to show something that is nanoparticle. So this is it. I know it's, what happened is that this cell, for example, has certain specific protein on top, and this is an antibody that recognizes this protein, and we attach to it. At the end of that, there's magnetized beads. I know one of my next speaker will talk a little bit more about magnetic particles, and because you attach to that, you put it through a magnetic field. Any of those cells that don't have this antibody or attached to will just elute it out, and therefore when you release the magnetic field, then all your cells that have this antibody with the beads on top will stay behind, and that's the principle that we apply to the clinical trial. Now, the outcome, almost finished now. The first thing is that clinical trial is very important. The first thing you want to do is do no harm. So the first question we ask is that, did it kill any patient? Not cure, but kill. All right, and we have no death or no adverse effect. And the second very important thing that I want to mention is that every single clinical trial has gone through the what we call the Human Ethics Committee and to get approvals. And that's a very important part, because there's a lot of charlatans around the world opening up stem cell laboratories and clinics and claiming all sorts of things, and they are sharks. And the things we found is that G-CSF actually, by injecting the G-CSF, can improve the patient's performance. The second point is that we want to mention is that whether we use purified or unselected stem cell, has no enhancement on the effect of injecting the protein itself. And this is one of the lessons I want to mention. I show you an example of good results, and I show you an example of clinical trials where the result is unexpected, and emphasizing the importance of that. And therefore, my last slide, I just going to say is that the stem cell therapy has a very exciting future because there's more and more advancement occurring, and it has application in what we call the jargon now, the individualized medicine or the generic type of medicine, using either somatic cells, such as reprogramming of adult cells, what we call the iPS technology,